Today's topic, SC Framework, the Normalization and Batch Correction Notebook. If you haven't watched the previous videos, we recommend you to go back and watch them first. In this notebook, we focus on the normalization and batch effect correction of the data. Similar to quality control and filtering, this step is aimed to prepare the data to facilitate better results in the following analysis steps. However, with normalization and batch effect correction, the aim is to refine the data points in a way that the comparability between, for example, samples is enhanced, the influence of outliers is mitigated, and variances introduced by technical or otherwise unwanted sources are omitted from the dataset. As with the other notebooks, we start by loading all the required packages and the configuration file. To continue with the analysis, we first load the anndata_2 file, saved by the QC notebook, and look at the basic overview again. Then we continue by filling out the general input required by the notebook. The general input comprises 4 parameters. The species parameter, which we set to zebrafish. The group by parameter, which refers to a column name in the .obs table by which the data should be grouped. In our case, this is set to time point. The threads parameter is used to set the number of threads used for multiprocessing. And lastly, the batch_column parameter, which also refers to a column in the .obs table containing the batch information of the dataset. In our case, the column is called batch. There is one important note on the batch correction. Ideally, you would have multiple replicates per time point, which are sequenced in a way that replicates of different time points are in the same sequencing run. However, this is not the case in this dataset. Consequently, it is not possible to correct the time points on sample level without risking to lose biological variability. To still correct for technical variance, we decided to treat the independent subdatasets as batches. With the general input set, we examine highly expressed genes and determine whether to include them in the normalization process. Highly expressed genes are those that account for a significant proportion of total counts per cell. They are often associated with core functions of the cells and thus not beneficial in the following analysis. Highly expressed genes can be excluded from the normalization process since they may exert a strong influence on normalization results. The plot reveals that a few genes are highly expressed in the dataset, leading us to exclude them from the normalization process. This exclusion does not imply removal from the dataset. They are simply omitted from the normalization calculations. Next, we perform the normalization. The counts for each cell are normalized so that all cells have the same number of counts after normalization. This removes imbalances due to sampling effects or sequencing depth to make the cells comparable. To normalize the data, we use a combination of the scampy functions normalize_total and log1p. Hereby, we aim to remove variability introduced by the sequencing depth by normalize_total, which is basically a scaling resulting in values between 0 and 1. The log1p function removes outliers by taking the logarithm of the data. Following the normalization, we predict the cell cycle phase for each cell derived from experimental observations. This information can be valuable for interpreting downstream analyses, particularly when observed patterns align with variations in cell cycle phases. The function generates 2 stacked bar plots, one displaying total cell counts, and the other depicting the percentage distribution, each per group and phase. In single-cell RNA sequencing, highly variable genes are genes whose expression levels show significant differences across cells. Identifying highly variable genes is a critical step in the analysis pipeline because they help distinguish biologically meaningful variation from technical noise. They highlight genes driving cellular diversity, such as markers for rare cell types, developmental states, or disease-specific signatures. By focusing on highly variable genes, downstream analyses like dimensionality reduction and clustering better resolve distinct cell populations, and they are used to identify genes associated with cellular transitions or disease progression. To identify highly variable genes, we use the scampy function highly_variable_genes. There is a variety of options for this function, but we recommend using the default parameters for now. The generated plots represent each gene as a single dot, with highly variable genes visually emphasized. The x-axis displays the mean expression level, while the y-axis shows gene dispersion, reflecting the variability of gene expression across all cells. The plot should resemble the example provided here. Highly variable genes typically exhibit high dispersion combined with moderate mean expression. Another important property of our data is its high dimensionality. However, this complexity hinders in-depth analysis, for example, the identification of cell states. Thus, dimension reduction algorithms are applied to reduce complexity while simultaneously retaining patterns, a crucial step to enable embedding and clustering. In other words, the data is organized into principal components, or PCs, of shared variance, so that noise is reduced by removing low-variance PCs, as well as PCs explaining technical or otherwise unwanted factors. This has also the benefit of reducing the computational demand. The upcoming heat maps and bar plots help identifying potentially unwanted PCs by showing them in combination with available cell-related metrics and gene-related metrics. In general, selected PCs should avoid any correlations, but the importance of metrics and the stringency of thresholds depends on the experiment and the underlying questions, and therefore requires careful consideration by the analyst. As a first step, we select the number of PCs to compute and the default metrics for visualization in the PCA plot. Some of these metrics were saved in the anndata.uns section during the QC notebook. We choose the default metrics, which are displayed by the following cell. Next, we run the principal component analysis, or PCA, a dimensionality reduction algorithm, for which we only consider the previously identified highly variable genes. The algorithm reduces the data's dimensionality to the number of dimensions set in the n_PCS parameter, with each dimension referred to as a PC. For the following step, 2 additional parameters have to be set: the number of PCs to display in the correlation heat maps and the metrics to visualize. By leaving the PC_color parameter empty, the default values are used. The plots generated by the subsequent code cells represent a crucial visualization in the analysis pipeline. They visualize the first 2 PCs, where each dot represents a single cell, with colors corresponding to user-defined metrics. These visualizations reveal potential biases or unintended correlations in the data that require mitigation. For instance, does total cell count correlate with a PC, or is there a relationship between mitochondrial gene percentage and the PCs? Additionally, assessing batch distributions in these plots helps determine whether batch effect correction is necessary. Let's take a look at the PCA plots. Visually, total counts and genes per cell appear evenly distributed, though a few clusters show low counts. Mitochondrial content strongly drives one cluster, while ribosomal gene content is generally high except for a small bottom cluster. Predicted cell cycle phases appear relatively uniform in distribution. Notably, batch visualization reveals a subtle displacement of cell clusters across batches, particularly in the bottom right cluster. This spatial displacement across batches often indicates the presence of batch effects requiring correction. To refine interpretation, we examine correlation heat maps comparing the top 15 PCs against both cell-level annotations and gene-level features. The key observations for the cell-level annotations are that PC1 shows strong correlation to mitochondrial and ribosomal content and moderate correlation with splicing and total counts. PC2 and 3 show moderate correlations with mitochondrial content and cell cycle, and PC5 demonstrates moderate correlations with both ribosomal gene counts and the S-phase score. The gene-level features show minimal correlation overall. Next, we select which PCs to remove before proceeding with downstream analysis. PCs heavily influenced by quality metrics or technical biases should be excluded. Retaining these PCs risks distorting downstream UMAP visualizations, where observed structures may reflect undesired technical artifacts rather than biologically meaningful differences. However, overfiltering PCs risks eliminating meaningful biological variance, leading to loss of critical information. In the next step, we generate a bar plot to aid you in the decision of which PCs to keep and which to remove. For this bar plot, the notebook provides additional options. The subset_PCS parameter lets you decide if you want to remove any PCs or if you want to keep all. The CORR_thresh parameter sets the highest absolute correlation that is allowed. The perc_thresh parameter lets you choose the top percentile of PCs that should be kept. The filter_methods parameter expects a list on which the PCs will be filtered. By default, this is set to both the variance and the correlation. The basis parameter lets you choose if the correlation is based on the cells or the genes. With the ignore_calls parameter, you can set columns that should not be included in the correlation computation. For this analysis, we stick to the default parameters. As all correlated metrics originated from cell annotations, we leave the reference source as OBS. In the bar plot, the proposed PC subset is displayed with darker bars representing the selected PCs. Based on the selected filter_methods, a vertical and horizontal threshold line is displayed. PCs are filtered if they are below the horizontal threshold for the correlation threshold, or if they are to the right of the vertical threshold line for the percentile threshold. With these selected parameters, the notebook proposes a list of PCs to be kept. This list is not final and can be adjusted by changing the following blue input cell. While not perfect, the initial PC set is a good starting point. Despite high correlation with mitochondrial counts, we expect a cluster of high mitochondrial cells due to the heart tissue origin. But selecting the PCs often involves an iterative refinement process, where analysts may need to revisit earlier steps to refine the selected PCs based on quality metrics or biological relevance. In our analysis, manual adjustments led to a final selection that diverged significantly from the automatically generated recommendations. The bar plot is then plotted again with the final set of kept PCs highlighted by the darker bars. With the last cell of this section, the PC filtering is applied. With the PCs filtered, we construct a k-nearest neighbors or KNN graph connecting cells based on the selected dimensionality reduction components. This graph captures the data structure and is used to identify clusters visualized in the UMAP in later steps. The number of neighbors can be adjusted. As described in the SCAMPy documentation, the size of the local neighborhood, in terms of number of neighboring data points, is used for manifold approximation. Larger values provide a more global view of the manifold, while smaller values preserve local data structures. Values are typically in the range from 2 to 100. For this analysis, we stick to the default of 15. Lastly, we come to the batch effect correction. Batch effects are variances in the data that are not intended by the experimental design, for example, technical variances. They can be introduced through various sources. For example, sequencing samples at different time points may introduce batch effects. As batch effects could interfere with downstream analysis, they are typically removed. However, it can be challenging to identify and correct for batch effects, as this is highly dependent on the experimental setup of the dataset. There are several batch correction methods available, which may perform differently depending on the dataset. Therefore, an overview will be provided to compare batch correction methods and select the best performing one. To help in the decision-making process, multiple methods can be included in the overview along with the local inverse Simpson's index or LESI score, which explains how well the batches are mixed after applying the correction. The input field lets you select which of those correction methods should be included in the overview. The SC toolbox supports the batch correction methods: BBKNN, MNN, Harmony, Scanorama, and Combat. For further details on those methods, visit our documentation. Specify the methods you want to use in the batch_methods parameter. If you want to skip the batch effect correction step, you can do so by setting the perform_batch_correction parameter to false. For this tutorial, we use BBKNN, MNN, Harmony, and Scanorama. The next cell then runs all correction methods and saves the resulting corrected anndata objects along with the uncorrected version in a dictionary. Next, the Uniform Manifold Approximation and Projection, or UMAP, is calculated for all corrected datasets. UMAP is a dimensionality reduction technique for visualizing high-dimensional data in 2D. For each plot, we set metrics to visualize using either default or custom lists. You can also choose to perform preliminary clustering to compare method performance. In this case, we take the default metrics and add time point as an additional metric. The next cell then runs a preliminary clustering using the Leiden algorithm, if enabled. After clustering is calculated, the LESI score can be used by measuring the heterogeneity within a local group. Comparing the lazy score between uncorrected data and the batch correction methods can help in deciding which method performed best. The lazy score, stored in anndata.obs, indicates the effective number of different categories represented in the local neighborhood of each cell. If the cells are well mixed, then we expect the lazy score to be closer to n for a dataset with n batches. The higher the lazy score, the more mixed the batches are. But while lazy scores can guide selection, they are not always decisive. Evaluate batch mixing and UMAP quality to choose the best method. With the lazy score calculated, we now plot the overview to compare the different batch effect correction methods. The overview includes selected metrics in PCA and UMAP and clustering, if enabled, and a box plot showing the lazy scores. Each column represents a different correction method or the uncorrected data. Here you can see the PCA plots from each correction method colored by the selected metrics. Here you can already see a difference between the methods. bbcann does not change the PCA due to it replacing the cann. The following rows show the PCA plot colored by the individual batches, the time point, and show the preliminary clustering. From the batch row, you can see the differences, except for the bbcann version. Overall, from those plots, it looks like batch effect was sufficiently corrected in all versions. Further rows in the plot show the individual UMAPs, also colored with all selected metrics. The UMAP now lets you also include bbcann to the comparison. In the last row, you can see the lazy scores. Since one batch makes up most of the data, the lazy score does not help a lot in this case. Optimally, each batch is similar in size. From the UMAP, we decided against bbcann and MNN. For MNN, the clusters are too separated by the batches. As for bbcann, you get less distinct clusters. In the end, it was between Harmony and Scanorama. We chose Harmony due to its robust performance compared to other methods. Select the final method used in the input cell. The following cells overwrite the current and data object with the corrected one. Lastly, as for all notebooks, you are given an overview of the final and data before it is saved to an H5AD file. With that, we have completed the third notebook. If you plan to use this or one of our other notebooks, please check out our Docker image, which comes as a full functional environment for the SC framework to be used within a web browser. In the next notebook, we will focus on the dimension reduction and on the clustering. If you need further assistance or want more information, be sure to check out our documentation or visit our GitHub repository. Thanks for watching.